Well, welcome everyone. Welcome to this Genomics England supply event. It's nice to see so many of you here. You all know the form for supply events. Every 10 minutes, I'll ask everyone to stand up. We move 3 chairs, and then you'll sit down again. No, it's not like that. It is actually the supply event for annotation, and as I look at the huge challenge we have in Genomics England to sequence and put together with the clinical data 100,000 whole genomes, this is the area where there's most for us all to learn, and we hope to learn it from you. So this is a very important event for us. So if I can say, to start off with, you waving your hand in the air because you want me to speak louder.
I'm going to give you this, so John, please begin.
Right. So before we get going, I need to do the normal things. If by any chance the fire alarm goes off, then we're all going to have to leave suddenly and go downstairs and, well, I would say assemble out on the green, only there isn't a green, so have to assemble out somewhere not on the road. So that's what we're doing. Other than that, we have a schedule in front of us. All of us here in front of you now are going to be speaking one way or the other. Mark Caulfield is our chief scientist. Jim Davis, beyond there, is our Chief Technology Officer. Tim Hubbard is our Head of Bioinformatics, and Nick Maltby is the important person you need to pay attention to because he is our General Counsel. That's us. We'll be talking, and then we're looking for questions from you. As I said to start off with, this is the area of greatest uncertainty as to how to get the best out of the program we're engaged in, and the area in which we most want to talk to you as the experts in the field in order to learn how to optimize our program. As I'll be saying several times during this this session this afternoon, as indeed my colleagues will be reiterating, the thing about this program is we're doing it at a colossal scale, unequaled in speed and volume before. So we do not have time to hang around and do a lot of research as we go along. There will be opportunities to do that, but it will not be part of our program. Our program is involves buying from you the software which will run on our system and provide the annotation. There will doubtless be a number of software suites which we will purchase in order to do that, but that's the way it's got to work. It's got to work that as the sequences are done, they get annotated, they get put into the data set, they get used by the community. How we go about that is something which we intend to learn together with you. So, let me kick off. Do I press one of these things? Yep. Okay, let's get going. I'm going to just give you the quick introduction to Genomics England, what we're about. As you'll know, this program was kicked off by the Prime Minister last December. There was then a process of consultation within the department and the NHS, which led in July this year to the launching of Genomics England, and we've been on the road since then. What are the key features of it? I'm not going to go through them all because I'm sure that you're basically familiar with it. We're doing 100,000 whole genomes. We're putting that together with the clinical data, and we're making it available to the community to analyze. We are an infrastructure project, crucial Remember that, we're an infrastructure project. There will be plenty of people who will be using our data set to do the research, to learn the clinical insights, to use it to develop diagnoses and products, but we are an infrastructure project. Our aim is to provide that data set both for the community of users outside the NHS and for the NHS itself to use it for what we believe will be the revolutionization of medicine in the NHS in due course. That's what we're about. This is our program in outline. We are obviously at the start of that. We have already, together with Cambridge University, launched the first of our pilots in rare inherited diseases. And we have just started the process together with CRUK in common cancers. We've done that latter process by means of giving 15 sequences to any sequencer who feels that they can give us back whole genome sequences within a month. We are looking forward to receiving that back in a couple of weeks' time. And at that stage, once we have the sequences back, we're going to want to provide those to you to run through your annotation software to show us what you can do. There'll then be a process which we are still developing as to how we assemble the learning from that and decide on our next steps. And so we are very much interested in what your view is as to what you can produce from that. Now, I have to say that amongst the community here, there are really three opportunities that are available to you to engage in this overall process. One opportunity is we obviously need help to manage this process, and we're going to need experts to help us assess what we're getting back and make our decisions on the basis of it. And so there is an opportunity for groups of experts to provide us with that support. Clearly, if you elect to participate on our side of the boundary in this whole process, then you can't at the same time offer yourself as a software supplier into it or an annotation supplier to it. So that's one. The second is you can offer your annotation algorithms for us to purchase through our tendering process. Now, almost certainly we'll be having more than one supplier, depending upon the kind of specialisms that your software is optimized for. But that will clearly be a competitive process, and we'll want to choose the best that is available. We may well, probably will, add more software applications over time into our suite as more come available and show themselves to be really effective. So the initial choice may well not be the end choice, but that is certainly an opportunity, but we will be making an initial selection in the early part of next year. The third opportunity that you will have is to participate beyond Genomics England in the process of integrating the learning from our data set into the clinical community. And I think my next slide illustrates this. This illustrates our overall process. We gather the data from the samples from the NHS and the clinical data from the NHS. We run the samples through sequencing factories. I use the word sequencing factories amongst us in this room. just to emphasize the fact that these are robotized, high-volume facilities. We call them sequencing centers elsewhere because that's a more friendly term. But be in no doubt that these are very high-volume sequencing facilities which will be turning out 100 or so whole genome sequences per day, and therefore at our end of the process there's no time to stop and individually assess each one of those. However, the passing of that information back into the NHS will require absolutely an assessment of the individual sequences, the interpretation of them, and we imagine that in the NHS there will be a process of understanding, of getting teams of experts around them in different parts of the country, supported by a suite of tools, and there will be an opportunity for people from the interpretation community to engage in that process as well. And hopefully there'll be feedback from that back into our system in terms of new algorithms having been developed which appeared effective, new insights being found, and that then being reintegrated back into our standard processes. So those three levels on which those of you from the annotation community can participate. Underneath all of that, there is the business model which we operate, from which we will eventually develop our revenue streams, and that is the community that I've just outlined before will be interacting with us to buy sequences, buy access to the sequences through a pay for access model, or SMEs in particular might prefer to sign a royalty agreement with us, or we would be open for joint ventures in particular spaces, so our reward would be an equity stake, or there may well be customers who want a degree of exclusivity in certain areas of the patient community over a short period of time in order to give them a chance to achieve their ends. So that's what I wanted to say by way of general introduction. I will now pass on to Mark, who, although it says rare diseases, will actually be talking about the disease community, the patient community, which we will be addressing.
Thanks very much, Sir John. So I'm going to cover a series of slides around. Sorry. That's okay. We'll just wait for the slides to come up. But I'm going to cover a number of areas that are the core focus of Genomics England programme in terms of the health gains for the community, and also the potential wealth gains in generating new opportunities for diagnostic and treatment development with the NHS as a platform in England for that. So we have a focus, as you will have seen and heard, on rare inherited disease, common cancers, and also, in addition, pathogen genomes. And I'll go through a little bit about each of those areas and how we've been thinking about that, so that you're enabled in terms of our thinking regarding diseases. So that's it, great. So the agenda of the programme, as you will have guessed, is to transform the capacity and capability of the NHS to deliver genomic medicine to the health system in the UK, but also to provide a very large programme around which we can mature the technologies and the approaches to maximise the benefit for patients. So you've heard about the 100,000 genomes. Many of you will have wondered about why whole genome sequencing. The reason for that is that we can see evidence that the potential for using whole genome sequence to advance our understanding of rare inherited disease and common cancers is now, and the UK, in the form of Genomics England, has decided to seize that opportunity. We want to leave a legacy of infrastructure that will support this being a continuing feature of NHS care in this country. So although we will end our mission to do the 100,000 genomes at the end of 2017, we see this as creating a legacy of infrastructure alongside which we will place and plan an education program to deliver a new cadre of people who will be able to provide the human capacity to deliver this within the health service, but also, in addition, the human capacity to support the activities of your companies or research group if you're from academia. And the Prime Minister's ambition is not at all modest. It's for the UK to be the world leaders in genomic medicine, and nothing less will do. And I recognise some of you are not from the UK, but with this £100 million support base, we believe there's a major opportunity to make a substantial impact. So let's focus on the specific diseases. So rare inherited disease is a focus because although these diseases can affect, depending on how you define them, up to 7% of the population, so by some definitions, EU definitions, 5 per 10,000, there are over 7,000 rare disorders, which are often disabling, usually shorten life, and they're extremely expensive. And for very many of these, although we anticipate that about 85% have single gene defects, only 600 currently, or 700, have actual testing going on within the National Health Service for these disorders through the UK Genetic Technology Testing Network. These represent about a quarter of the known disease genes. So we still have not got significant penetration. And if you look on the web at the Orphanet report around how many gene tests have been adopted in other European countries, you will see that the UK is relatively modest in its adoption of these tests compared to European neighbours in Germany and Spain. This is also an area of where there is the potential for repurposing of medicines, the potential to develop new medicines, and there are exemplar medicines that are under trial currently in the United Kingdom throughout the world that are derived from rare inherited disease programs. And certain changes to the way that tariffs are viewed, tax breaks and other things, mean that this is a significant opportunity to develop medicines for disorders for which actually, frankly, there often is nothing right now. In common cancer, you will have seen the advisory group reports on these areas on the web, on our website, and if you haven't, you can go to our website and download those and look at them. We have got a focus on common cancer and also on rare cancer. The reports gave a steer towards looking at lung cancer, and in our current pilot program we've already broadened that out, and I will be emphasizing what the pilots contain in a moment, but suffice it to say that our pilot in cancer will be lung cancer, breast cancer and colon cancer, and we will be doing that over the course of next year. We will also be doing a rare disease pilot, and I'll flesh that out for you in a moment. The lung cancer steer is because lung cancer has got a terrible outcome, and it also is an area where stratified medicine has already gained traction. So, for example, knowing the genetic architecture of the tumour allows us to select therapies. Imagine a situation in which we could use other therapies in a combination based on the evolution of that tumour's genetic architecture over the life of the cancer, and potentially convert the common cancer from a disease with a very short lifespan into a chronic disease, much as we've done, say, for example, with HIV and some other chronic infections that were previously largely fatal within a short space of time. So to emphasize to you that we haven't chosen the cancers that we will focus on for the main program, nor have we chosen the rare inherited diseases as yet, but we will be looking to the NHS to provide us with the flow of samples to generate the opportunities there for patients. We will also have some focus on rare and childhood cancers, particularly because in childhood cancer, if you are a childhood cancer survivor, you have in certain cancers an 11-fold risk of more premature mortality because of other cancers or because of debilitation as a result of the treatment. So this emphasizes why lung cancer might be important. Although it's got a similar cancer incidence, as you'll see up the side, the outcome along the bottom at 5 years is far worse for lung cancer, probably in some measure because of its late presentation, but also because we have a potential to stratify care. There's an opportunity for us. We also have a mission to focus on pathogens, and here we're working very closely with Public Health England and the reference laboratories who already do some of this work. We have a steer to look at HIV, hepatitis C and tuberculosis, but to emphasize why we might be looking at those particular infections, it is around the elements within the pathogens that cause resistance. For example, for HIV and hepatitis C, a knowledge of the mutation architecture of the organism may allow us to select therapies and tailor them to the individual more effectively, so we do not expose the person to side effects without any potential for benefit. We can also use this technology to track outbreaks and also to work out where outbreaks originate from. And this is actually can be quite a challenge. This is very useful to prevent organisms from resurfacing after a period of apparent dormancy. So there are several routes by which this can be used. So my emphasis to you today is that there are substantial potential health gains from this. You've already heard a lot about this program. This is for the NHS, by the NHS, and the benefits will be for patients, but the benefits take two formats. By working with the NHS, our academic community, and you as industrial partners, supporting that with education, we should be able to leave a collective legacy of next generation centres, sequencing centres, a sample pipeline, and a biorepository and large-scale data store that makes this the route to go forward with genomic medicine worldwide. And if we do this here, and if we do it together, then not only will we have made the UK, and I realise some of you aren't from the UK, the best place to do this, but the ramifications for this programme, for other parts of the world, and for adoption by other Western governments will become a must-do programme. And we only have to look at funding patterns from the announced call for Horizon 2020 to see the priority that this type of aspect has already in the European Union ministers and governments across the Union. So this is a priority for all of us wherever we are, and that's also true in the United States and in other countries in emergent economies. But we do need something from you, and that is because this data at its full deployment will be a hose pipe of data coming out the end. We need you to probably work together with us in a way that you mightn't normally do as companies to actually deliver a really high fidelity product so we can use this program to produce the highest probable outcome for patients and the highest benefit. And if you do that, I have no doubt that for those of you who provide high quality, high caliber products, there will be benefits from the capability to market that in other areas. So the opportunities from this are new diagnostics and new therapies for patients. And we're going to do it all by the end of 2017. So my question is, are you ready? Because I hope so.
Thanks. Change the slides. Nope, not that one. There's a clicker.
Oh, sorry.
Yeah, sorry. That's smart. Okay, so I'm going to talk about annotation, which is what you're all here for today. So this is the pipeline, the overall pipeline of the whole project, so going from samples all the way through to clinical action. And you'll see there's a slightly larger dotted line bit in between variants and clinical interpretation, which I'll come to. Now, the way we're arranging this is to split it up into separate components. We're not looking to procure from a single entity all of those steps. So at the moment, we're engaged in the sequencing part. We're doing a bake-off with a small set of samples, as John mentioned, and they're going to generate clearly defined BAMs and VCFs. What we're interested in getting from you is the next bit. Now, that might just be interpretation of VCFs, and to take that and turn it into sort of candidate variants. It may involve a degree of clinical interpretation on top of that. That's really a question that we're trying to find out what you can kind of do. Now, we're looking only to procure a software pipeline. We're not looking to procure actual annotators looking at that. We're assuming that that will happen on the NHS side of the world. So either a list of variants or the sort of automated interpretation will go back to people in the NHS, and they'll look at that, maybe do some validation, and then take clinical action on that basis. So that's the narrow pipeline that we're looking to procure. Now, the output from that won't just go to the NHS, it will also go into our database. So that's the research database that will have the BAMs and the VCFs available for research that Jim's planning to build. But it will also have this annotation in there as well. You know, the variety of people likely to use that resource. A lot of people want to run their own pipelines to do their own analysis, but to have a baseline from the pipelines that we're using will be useful as well, particularly for some people who don't have that capacity. So that's the overall outline. Now, the sequence assessment. So the Bakeoff's in progress. So a small set of samples are with suppliers, and we'll be evaluating that on quality and coverage. And that's fairly well established how you go about that. It's not trivial, given that the sequences are very different from different suppliers. You know, it's difficult to work out the various metrics, but you know, it's fairly well defined. Now, annotation assessment is a much harder problem, as I'm sure you're all well aware. So one problem is simply the lack of the gold standard of what's the right answer. We're going to be talking here about individual samples where it's not clear exactly what's in that sample. We may have a clinical diagnosis, but there'll be other things in there that may be, you know, other findings which you might discover and not know whether they're right or not. So establishing, you know, evaluating these samples, even if they're very clean, is going to be quite hard. The other thing is there aren't really so clearly established data standards. So in the sequencing side, it's fairly easy. We have BAMs and VCFs, you know, the specification. In the case of what's the output from annotation pipelines, we can make a ranked list of variants, but then there's the various phenotype descriptions of that, the ontologies around that, what's the clinical interpretation look like? Those are all fairly undefined. And then there's other types of associated indications that some of you might want to provide on top of that. So now, there have been lots of exercises in the past to evaluate things. You'll be familiar with some of these, not all of them in this particular space. The most recent two at the bottom, though, Clarity and KG, clearly are in the game of trying to understand variants. Now, some of those, they're set up in a game in a certain sort of way to answer a particular scientific question. Does an algorithm work out a particular answer from a particular designed question? Some of those are kind of artificial. They're not quite real-life situations. So what we're going to give you is a real-life situation. Well, obviously, because we want to end up with a pipeline that we can use in a real-life situation. So you're going to get sequence just as you will get it in the real-life case, BAMs and VCFs, trios for rare diseases and cancer germline tumor pairs. And you'll get the phenotype data that we have, that we'll have in real life, like what's available to clinicians. So that may or may not be particularly tidy, but you'll get what is available in the sort of health service situation. What are we anticipating to get back from you? Well, obviously the most obvious thing is a ranked list of variants with estimates of pathogenicity for some of those variants, confidence of how good you think your estimate is, then higher level analysis potentially, so how that impacts particular genes or clusters of genes or pathways, underlying molecular mechanisms or contributions to disease. Some of you may have tools that organize the literature to make it easier for the sort of subsequent clinical interpretation within the health service to look at things that are relevant, maybe hopefully organized in a way that isn't very time consuming to use. And particularly to that end, we're looking for clear clinical reports. Again, thinking from the health service point of view, they don't have a lot of time to look at these things. They want to get as clear, simplified picture as possible, with the opportunity to go and borrow if they are specialist clinical geneticists, for example. And some of you may suggest clinical interventions. So you may be mapping onto, say, for cancer, the particular drugs that you should select given the defects in particular genes that you've uncovered. I think one thing to say is that the VCF files, they contain the data that's been extracted from the sequence, so where the CNVs are, where the variants are. We're asking for what the meaning of that is. So you might want to recall the VCF. Some of you may want to do that. You may feel you've got a particular pipeline, but we're interested in what the meaning is. That's the output that we're really going to evaluate you on. So the kind of assessment criteria: accuracy, how clinically informative your output is, how fast you can turn this round, how understandable the output is. We're going to expect standardized data outputs, and we'll move towards some standard over time. And more importantly, the ability to operate at scale. So even in the pilot, it's going to be at the level of 25 samples a day. And that will scale up through the main project, 2015 to 2017. So this isn't going to be enough time to go and look at things. You're going to have a pipeline which is automated and can just run through. There's another, right, so this is how we're going to do it as far as we've worked it out so far. So we're going to get these samples back from the sequencers. Obviously we're getting it from multiple sequencers. We will pick the one we think is best based on our initial analysis, and those will be the ones that will go back to you, the ones who want to take part in the evaluation. And we'll then really investigate the kind of levels of information that you're going to provide. At that stage, there'll be a minimum requirement for file formats. We'll really probably just want you to have a list of variants as a sort of single sort of spreadsheet type output as part of the analysis, but obviously we'll be looking at the sort of your particular specific things that your particular offering is generating, and we'll use that to inform our future specification of common formats. And so from that, we'll select multiple suppliers for the pilots. And if it turns out there's too many of you that pass that, we'll probably have to do a more sophisticated analysis with more samples to evaluate who's best to select, because we can't pick large numbers. So we'll pick the suppliers partly on the ability to deliver timely, consistent data sets, and it's not clear everybody will be able to do that. There'll be ongoing assessment during the pilots, refining the file formats, and then at the end of the pilot or during the pilot, we'll be tendering for the main program. So another thing that is worth saying, so the pilot is slightly different in terms of the samples, the consents are different, the requirements to return in real time to the health service are different. During the pilot stage, whatever setup you've got, we can probably work with that. But during the main program, we'll be expecting to run all this in a data center behind a closed firewall. So there won't be any sending sample data to you. You're going to have to provide a virtual machine with your infrastructure on it, which will have to run inside the data center. And you can also expect, just to mention, if you've seen The Economist in the last few weeks talking about reproducible science and the lack of it, there's going to be a requirement for reproducibility and evaluation and compliance with, at least showing what you've done in terms of your change processing, and proof that you haven't screwed everything up and made the next version not work at all. This is kind of like the sort of clear dry lab sort of certification, which is beginning to be discussed in the US, and the regulatory agencies are we're going to have to live in that world, or some aspect of that world, if we're going to produce clinical decision software. And that's it. I'll pass on to Nick.
So Nick, you missed a speaker, Chairman.
Sorry, we're out of place.
Don't leave me out.
Although I was just reading.
Although that said, Tim has just made the two key points that you would be interested in terms of the data and IT infrastructure. So you know what we want, pretty much. You say you're ready. I saw some people going, yeah, when that question was asked by Mark. We need to know what you need. If you look at software engineers, they want to know your requirements. They want to get as much information out of you as possible before they have to take design decisions. So they don't like taking design decisions and then finding out that they have to revisit them. That's not good. It's expensive. So you've got the two main constraints. The idea is that the data is going to stay within the NHS. So we're not sending VCFs off to people and expecting annotations back. The other one that Tim was edging towards with the CLIA mentioned, CLIA. Really what we're interested in is software that plays nicely. Now, we've heard a lot about the legacy of the program. That's great. One thing that isn't improved when you call it legacy is software. What we're most interested in here is software that plays nicely. We understand the quality of the software that, obviously you'd have modern software processes. If you're producing software, you'd understand, just basically change control is the simplest way of looking at it. We need to, and accountability and a degree of transparency. So of course you might have some special sauce inside, but we'd expect you to have that fully documented and accountable. If people are going to use your software, then we need to know how, what version of the software is being used, what configuration was used. It needs to play nicely in terms of telling the environment it's running in about this, or you need to provide some suitable interface, so that we have more information about provenance for the data that we finally provide to support clinical decision making. It's going to be pretty important. So that's really, I'm just here, here to listen for what you would need to do the best job you could do for us. That's about it, really. I'm here to answer questions.
Thank you. I'm just going to spend a few minutes putting some flesh on the bones of what we've said this afternoon. Because obviously, clearly, you're here to sort of get a feel as to how you get involved with our program and what our dates are and what timelines we are working to. So just 3 slides. The first slide is essentially, I've sent you the sequencing assessment questionnaire, which we've touched on. But we launched the sequencing assessment exercise at the beginning of October. We're expecting to get output from that. I think it's I can't see that far, but I think it's the 24th of November, 25th of November. So we will at that point have. We've talked about we've got 15. We sent 15 DNA samples out. We'll obviously get 15 reports back from multiple providers. As Tim has said, we're going to select the best of those, and obviously not on the 25th, but probably when we've done some validation of what we've got back to make sure that what we send you is meaningful. And so I think we're probably looking at 3 weeks after that. We will be in a position both to release an annotation assessment questionnaire, and I suppose more importantly to release some BAM and/or VCF files. And obviously I've had some conversation with some of you in this room already as to what you think about which of those you need. And clearly, also the purpose of this event is to get feedback. So either in the questions that follows this, or after this, or indeed any time after this event, please do come back to us and let us know your views on some of the things that we're talking about today. So going back to this slide, we are looking to make sense of the sequencing by 10th of January, and we will have a view as to who is above our line and who effectively can participate with us in our cancer pilot. And this is 3,000 germline and 3,000 tumours. That's 3,000 people, and we will be tendering that starting probably very shortly, with a view to contracting for that at the end of probably March, and with first data output from cancer probably sometime in Q2 2014, We also have a rare diseases pilot where we're expecting some output, we hope in Q1 2014. So in terms of annotation and having reports available for annotation, we're going to have, certainly it's going to ramp up through 2014, but certainly Q1 and Q2 we will have hundreds of reports available for annotation. And I suppose, I mean, given that these are pilots, they're pilots to the main program, we are hoping to learn as much as we can from our pilots and from the, from the outputs that we get, both from sequencing and annotation, because clearly we need the output to inform how we set up the main program. And again, according to our program, according to our calendar, we are going to be going out to tender for the main program probably in the middle of next year. We've already touched on some of these. I mean, the reports that we're going to get back on the 25th are cancer. They're not rare. So obviously the reports we send to you will be cancer and not rare. We have debated internally whether we do need some rare. At the moment, our plan is to send you the cancer, as it were, and to get you to do what you do with those. Sample size, I mean, we've got 15. We think that's probably enough, but I mean, as Tim has touched on, we might go for a lot more if we cannot separate, on the basis of 15. We need common standards. Turnaround time, again, I've been interested in views. How long, we've given the sequencers a month. Question, how long do you in the room think we should allow you? We want it to be reasonably challenging because we're ramping up. I mean, Tim's already demonstrated how many we're going to do probably every day. And the people we're going to partner with on the main program have to be, as Sir John has made clear, have to be able to work with us on a factory scale pipeline and to produce results. Evaluation we've talked about, and clearly, this, as we move forward, this is a comment more for the main program, but this is evolving technology. We don't want to be, stuck in a 2014 technological platform in sometime in 2016-17 when the world has moved on, and so clearly we want to work with people who are happy to evolve that technology with us. So, John, do you want to talk to this or small business?
Right. Well, neither you nor I are definitive on this, but for sure, what we're pointing to here is that we've been talking to the Technology Strategy Board, who have a parallel program to our program, and are interested in investing in this space. And it seems to us quite possible that it would be helpful to you as suppliers to engage in a program which they intend to run in the remainder of this year, to enable you to participate with us. I think one thing that we didn't mention up to now is that we are not planning to fund anyone through the evaluation process. We, Genomics England, are not planning to fund you to do your annotation as part of the evaluation process. But it is possible that the TSB will be launching a program which will enable at least part funding of that. They've put aside £10 million. Whether that's a sum that can be usefully spent in simply the remainder of this year remains to be seen. But we thought it would be worth mentioning to you that this is in the offing as a parallel activity alongside ours. It seems to me to make sense because, as I said at the start, this is the whole 100 genome program is at the forefront of technology in every part of its activities. But perhaps in this space, we are actually also at the forefront of science, which makes it that much more complicated, with probably more for you to do on the supplier side to bring it to a point where we can use. And so the TSB's offer to work alongside us, it does seem to me potentially quite helpful.
And if I could just add to that, I mean essentially, on the current time frame on that particular competition, I mean, we're looking at the 2nd of December for launching the competition, and between now and probably in the next couple of weeks, we need to firm up what the competition is about. Now, it's clearly in the annotation space, and if we can talk to you probably after this event as to what you think is most useful in that space. I mean, on the slide we've talked about increasing the rate and quality of annotation and also the interpretive value of it. But there may be other areas that people think are equally worthy of inclusion. But clearly, this is another area where we would welcome feedback, but we need to get that quite quickly.
Thank you. I think that concludes the prepared material we have. And so now, if we just move on from that, click on. So now we're really open to questions. You can ask questions here in the room, which I hope you'll do, because we intend to learn a lot, and I hope you will too. There are these forms available for you to write down anything you're too embarrassed to ask openly. And you can even send us emails, and we will answer any and all of those. We will also be available, all of us, after this, to huddle in quiet corners around this floor to answer anything else you would prefer to keep secret from your colleagues.